Welcome back to a study on plant tissues. This is the second video in the series. So last time we looked at meristematic tissue, and today we're going to look at permanent tissues. There are three main groups: epidermal, ground, and vascular. When you look at the three main tissues, the permanent tissues, if we look at a cross-section of a root, you can easily see here from the diagram the three positions of these tissues. So dermal tissues on the outside, ground tissues underneath that, and vascular tissues in the center. That's the basic plan for all the organs in a plant. However, the orientation of each of those three is slightly different in each of the organs. And when we come to do plant organs in another video, you will learn where those positions are. So let's start with epidermal tissue. Epidermis, epi meaning outside, dermal referring to skin or surface. And so these are the tissues that are on the outside, the surface of the plant or surface of the organism. Their function is to protect and to cover. So obviously we find them on the outside of leaves and stems and roots. Epidermal tissues are only one cell thick. They're very, very simple tissues. What's important though is that there are no intercellular spaces between those cells. And that's a protective feature to prevent other organisms like viruses or bacteria or protists from gaining access to that individual. Your epidermal tissues are transparent. In leaves and stems, this is obviously vitally important because it allows sunlight to penetrate so that photosynthesis can take place. On leaves and stems also, the above ground portion of the plant, there is a waxy cuticle. Now, you will know that oil and water do not mix, and so the waxy surface of this on top of the epidermis, that cuticle, makes the plant waterproof. It prevents evaporation from occurring, which obviously is very important for plants in summer, particularly in Southern Africa countries where summer is incredibly hot and dry. In addition, in both the epidermis above ground and the epidermis below ground, there are further specialized cells called guard cells and root hair cells. And we'll learn a bit more about those in a moment. In your garden, if you have what used to be called an Australian cherry, a zyzigium, you'll be able to do what you see here in terms of pulling off the epidermis on a young leaf. So you're looking for a small young leaf. They're usually red in color. And all you do is you bend the leaf, try and make a gentle fold in it, and crack the epidermis, and then you can actually pull the epidermis right off, and you can see here the epidermis is in fact transparent. If we look at that epidermis now from the top, so from above, you will notice these funny structures. So that is one cell and that is one cell. So there are two guard cells that make up a stoma or stomata plural. So the stoma is actually the hole in the middle with the two guard cells around it. You can see their shape is quite different. They have a sort of kidney shape. And what happens is that as the plant photosynthesizes, the water potential in these cells will change. So in your epidermis, these are the only two cells that have chloroplasts in them. Okay, all the green here, this is the tissues underneath the epidermis. The epidermis itself is not green. But your guard cells do have chloroplasts. They're the only cells in the epidermis that have chloroplasts. And when they photosynthesize, the water potential will change. Additional water therefore flows into these cells with the result that they bulge even more, which opens up this hole, the stoma, in the middle. And that stoma is used for gaseous exchange. Remember I said to you just a moment ago that epidermal cells do not have intercellular spaces, but the plant does still require oxygen, and it does need to get rid of carbon dioxide. If it's photosynthesizing, it obviously requires carbon dioxide as well, more than it can produce through its own respiration. And so gaseous exchange has to take place with the outside environment, and it does that through this stoma. When photosynthesis is not taking place, then obviously the water potential of the cells changes again, and it will be the same as the surrounding cells, with the result that these cells do not bulge because there's no extra water in them. And so they close, and that stoma that was visible in the middle over here now is completely closed up. Some plants are quite clever in that they don't photosynthesize during the day. When you learn about photosynthesis, you will learn that there are 2 stages in photosynthesis, one which is dependent upon the light and one which is not dependent upon the light. And so what the plants do is that they don't do gaseous exchange during the day. They close their stoma because it's too hot and dry. So you'll see this often in cacti and other plants that live in desert or semi-desert environments. And instead, they do the light dependence phase initially, and then the light independent phase where they don't require sunlight. They do that at night, and then they will open these stoma and do gaseous exchange at night and store up all the oxygen and carbon dioxide that they need for the following day. All right, if we move now to look at the epidermis in the root, if you've ever pulled a sapling or a seedling out of the ground, you'll see this kind of furry bit on the bottom of the root. Those are teeny tiny root hairs formed from root hair cells. So here you can see this is our epidermal layer. No intercellular spaces there, single layer of cells. Notice there's no cuticle on top of that, because obviously the function of the root is to absorb water, so you cannot have a waxy cuticle on it. And here you can see a root hair cell, and all that the root hair is an elongation on the surface of that cell. So it's not a hair like you and I have hairs that are made of keratin. This is the cell membrane that continues and spreads out, and the cytoplasm fills into this space, and this just keeps growing and growing and growing and elongating. And so here you can see these elongated root hair cells, these teeny tiny little fuzzy bits. The purpose of the epidermal root hair cells is to increase surface area. So you can see here's your cell membrane, and it just extends. So all of that is cell membrane, which means that absorption from the soil, whether it's water or minerals or whatever, can occur all along that surface, not just across the small surface. So if you look at that cell, it's only got a small surface area that's in contact with the soil. Here you've got a very long cell surface that is in contact with the soil, and so it increases the rate of absorption into the root. All right, ground tissues. Parenchyma are probably the most predominant ground tissue, and there is a specialized type of parenchyma that we'll talk about in a moment called Chlorenchyma. So the function of parenchyma is that it is a packing tissue. It fills up space, and it has a pretty important function in that when it is full of water, when the vacuole is nice and full, we call that condition turgid, and that turgid condition supports the plant. You may know from physics, for every action there is an equal and opposite reaction. So when the water in the vacuole and in the cell pushes the cell membranes up against the cell wall pushes back against the cell membrane, and it's that counter pushing that keeps the plant upright. Remember, a plant doesn't have a skeleton like we do, and so the only way that it can grow against gravity is if it has some kind of support and stability, and it gets that from being turgid. The opposite of turgid is flaccid, and flaccid means when a plant does not have enough water to support itself. And what we see on the outside is that the plant will wilt. So when a plant looks all floppy, it is wilted, and that is because the cells are flaccid, opposite of turgid. Ground tissue is also responsible for gaseous exchange. Ground tissue or parenchyma has large intercellular spaces, and in the leaf particularly, and sometimes in the stem, those intercellular spaces are filled with air rather than with fluid, and so gaseous exchange can occur. I said to you there's a specialized form of parenchyma called Chlorenchyma. So when parenchyma have chloroplasts in them, they're able to photosynthesize, and we'll look at 2 examples of that in a minute. Your parenchyma cells, your Chlorenchyma cells, they have very, very thin cell walls with exceptionally large vacuoles. The vacuole is the largest organelle in these cells. They are isodiametric, which often helps with when you're looking at a micrograph with identifying them. Iso meaning the same, diametric meaning diameter. So the width and the length of the cell are roughly the same. And as I said to you, there are intercellular air spaces or intercellular spaces filled with fluid, depending on where it is, and they're found throughout the plant. So if you have a look, top left is parenchyma, bottom right is Chlorenchyma, and they are roughly isodiametric, same top and bottom in terms of the diameter. Now, when we come to study the leaf, you will learn about two types of Chlorenchyma. We have palisade cells and spongy mesophyll cells. You don't need to worry about that now. We'll look at it when we do organs, just to mention it. And what we're seeing here in this bottom diagram, so here we have our guard cells. The stoma in the middle is closed. Here is a massive air space just inside of the leaf. You can make out the cuticle just along the bottom there, that black line. Okay, that's your waxy cuticle. And these large cells, here's another air space. Okay, this is actually air space, that's air space. There's a little bit of air space in there, another bit of air space in there. This particular tissue that we're looking at, this is your spongy mesophyll found in your leaf. All right, let's look at Collenchyma. Collenchyma, col for corners, Collenchyma, C-O-C-O, is the way that I try and remember this one. When you look at it on a micrograph, you will notice these thick, what appear to be black spaces between the cells. Those are cell walls, thick cell walls, and they have been stained, which is why this all appears very black. If you compare it, for example, here, if you look at the cell wall of that cell, and then look here at the cell wall of this cell, you can see the significant difference in the cell wall itself. And the function of Collenchyma, because it has such thick cell walls, is that it supports the plant. And so you find Collenchyma in stems and branches, and also in some leaves, depending on the leaf. If we look at a drawing of different types of Collenchyma, you can see quite easily the uneven thickening. So here you can see you've got very thick corners, not so thick in the middle. Thick corner, not so thick. Thick corner, not so thick. This is not just the cell membrane that has pulled away from the cell wall, as we might see in a plant that is flaccid. It is, in fact, this is a turgid cell, and this is, in fact, the thick cell wall in the corners. Even if you look at a more rectangular sort of cell, here you can see thin, just above the pointer, there's a very thin section of the cell wall, whereas here there is a very thick section of the cell wall. So the main point being that in Collenchyma, you have unevenly thickened cell walls, thickest in the corners. Sclerenchyma, and I know at this point you're thinking, why on earth do we have all these funny names? Unfortunately, you just have to learn them. Sclerenchyma is also got thickened cell walls, but its cell walls are thickened evenly. So the thickness of the cell wall is the same throughout the cell wall. It also provides structure, given the fact that the cell wall is quite thick. You can find this either in fibers or in kind of more rounder, or almost triangular type cells, which we call sclereids, or stone cells. Just for interest's sake, those of you who like eating pears and guavas, the slightly gritty, or in fact in apples, that gritty texture that you get, which gives you the nice crunch in the apple, and which you can obviously taste and feel in your mouth quite significantly in pears, that grittiness comes from the presence of sclereids, these stone cells. So if we look at a drawing of it, you can identify, so if you look here, all of this is thickened cell wall, and there's the cytoplasm in the middle. Okay, so there's your thickened cell wall, and if we draw it, here is your thickened cell wall, there is the cytoplasm in the middle. Now, it's important to note the word lumen. Lumen is what we refer to as the space in the center of the cell, and that's where the cytoplasm would be found. Now, obviously, if you've got a very thick cell wall, in this case, because the cell wall is evenly thickened around the entire cell, it makes it incredibly difficult for anything to move in or out of that cell. And so what happens is that you end up with these, what look like lines across. You can see them here, all of these black lines that appear, okay? And those are actually what we call pits. They are little tunnels, if you like, through the cell wall, and they allow transport to occur. So if we look at the structure of a sclereid in a little bit more detail, On the outside is your primary cell wall. That's the one that the cell will have initially as it's growing. And then when it reaches maturity and it's at its biggest size, it's not going to get any bigger, then it starts to produce these layers of what we call secondary cell wall or secondary thickening. Okay, and there's the lumen in the middle. Now, your cell wall, your primary cell wall, is made of cellulose, obviously, but also a little protein, a little molecule called pectin. Your secondary cell walls, though, all of them, they have cellulose, but they also have lignin in them. Lignin is what we refer to as woody. That's what gives plants a kind of woodiness, is the lignin. And so you find in these secondary cell walls the lignin, which gives it additional strength and additional toughness. All right, let's move on to our third group of tissues, our vascular tissues. There's xylem and there's phloem. Notice it's not xylem. Okay, some people who don't know how to pronounce this word would say xylem. It's not, it is xylem, like xylophone. There are two types of cells that we get in xylem. We get xylem tracheids and xylem vessels. Both of them have the same function, which is to transport water and minerals one direction, from the bottom up to the top. Okay, so it's going from the roots to the rest of the plant. What's interesting about xylem is that it is dead at maturity. So the cell starts growing, it starts laying down additional layers of thickening, exactly like our sclerids, our stone cells, and our Collenchyma. It gets this secondary thickening of the cell wall, but it becomes so thick that in fact the cell dies. When you have a tracheid, it's joined end to end, and I'll show you how that is now. It's sort of overlapped. When you have a vessel, it's literally just end to end. The thickening that you have, you get a variety of different types. The three most common types are annular, spiral, which is also called helical, and pitted. And obviously, because xylem is carrying water, you will find it everywhere in the plant. So if we have a look here, this is a xylem vessel, very large, and there's the end of the vessel and the start of the second one. You can see it's completely perforated. That's the word that we use, perforated, which means that the end wall has disappeared entirely. Everything that was inside that cell is gone. There's no cell membrane, there's no vacuole, there's no nucleus, there is nothing. The entire cell has died and been stripped on the inside, and all that is left here is the cell wall on the outside. When you look at a micrograph, this is what you would see, these massive, almost holes as they appear. Okay, those are your xylem vessels, and they are laid down end to end. Your xylem tracheids, on the other hand, are more pointed at the end. They're also thinner, and so they overlap with one another, and they develop these pits or kind of holes in the tips of each tracheid. So if this is our xylem vessel, then all of these little cells in between, these are our xylem tracheids. And if you have a look down here, you can see the xylem tracheids sitting close together. Obviously, this is a vessel, and that is a vessel. You can see quite nicely here the secondary thickening that has been laid down, which looks circular. So we've got annular, you've got your helical or your spiral, and you've got pitted, which is lots of little holes. Now the purpose of this thickening, particularly with the pits allow lateral movement of water. So they allow movement left to right rather than top to bottom. Likewise here with our helical or our spiral, we could have water moving through here and here and here. A little bit more difficult than that one where you've actually got a hole, but it will occur that way. So if we go back, so you can see what looks circular here is actually just half the picture, and the other half of the picture coming out of the slide would connect it in such a way that actually it has a helical arrangement. Now with phloem, it's not so much that we get two different types of cells, but rather there are two parts to phloem, or two components to phloem. So we have sieve tubes and we have companion cells. The function of your phloem, if the function of xylem is to carry water, then the function of phloem is to carry starch and nutrients. Now that starch and nutrients It could either be starch that has been stored in the root that during winter must then be broken down and transported upwards to the above ground portion of the plant, or it could be starch that is produced as a result of photosynthesis in the leaves, which must then be transported down to the roots to be stored for winter. So it transports starch and nutrients, and it transports it in two different directions, both up and down. Your phloem cells, both of them are living cells, but the sieve tubes have no nuclei. So most of the organelles in your sieve tube have disappeared. All that's left is the cytoplasm and the cell membrane. So how does the sieve tube know what to do? How is it able to carry out its function? Well, that's because it is controlled by a companion cell. Now, the companion cell has plasmodesmata that connect it to the sieve tube, which allow it to control the sieve tube. And again, obviously, because phloem is carrying both starch and nutrients into the roots and up to the leaves, it is found throughout the plant. So if we have a look at a diagram, here in the middle, that is one sieve tube. Okay, and you can see that the end walls are perforated, so we talk about a sieve plate, but it still has a cell membrane present. Sitting alongside is a companion cell. The companion cell has a nucleus, but obviously the sieve tube does not have a nucleus. And then notice these little breaks here. They're calling it a lateral sieve area, but the actual term for this little break here is your plasmodesmata. Okay. This is obviously looking at a longitudinal section. If we look at a transverse section or a cross section like this, here we've cut through our phloem at various points. For this one and this one and this one, we've cut through at or just above or just below, depending on which way around you're looking at your slide, this sieve plate. And so you can see the perforation, and that's a characteristic that you need to look for if you're looking at a micrograph or a scientific drawing. That is a dead giveaway that what you're looking at is phloem. Another giveaway that you're looking at phloem is the presence in between these large cells of teeny tiny little cells. And those teeny tiny little cells are your companion cells, these ones that sit alongside and control what the sieve tube is doing. If we look at micrographs, so here you can see these are the sieve plates with their perforations. Okay. And when we look at it in longitudinal section, there they are. You can see the little kind of holes going in. This purple is caused by dye that is being drawn up in the plant. Okay, so if we compare a xylem vessel and a phloem vessel, xylem is one way only from the roots to the leaves, whereas phloem is two-way, depending on the season and depending on what is required. Xylem carries water and minerals from the soil, whereas phloem carries nutrients, also in water, but its primary thing is to transport nutrients, sugar, for example. In your xylem vessel, there are no end walls. Obviously, in the xylem trachede there are, but not in the xylem vessel. Whereas in your phloem vessel, or vessel element, you do have these perforated end walls. So we've got a sieve plate. In the xylem, because of the fact that its function is to draw water up against gravity, there are very, very thick cell walls that have been thickened with lignin. So you have secondary cell walls thickened with lignin, whereas in the phloem vessel, it tends to be only your primary cell walls. If we look at a cross section, so here we've got a dicotyledonous stem, and again we will come back to the position in stems and leaves and roots when we do organs, but just to show you the different tissues that we've looked at today in situ, where they exist. So up at the top here is your epidermis, one cell thick, no intercellular spaces. On top of that is a cuticle. Okay. Some plants will have epidermal hairs. Those are for protection against grazing and infection and that sort of thing. If you look at this next layer, it looks like there's lots of black in between. Those are not spaces. That black is in fact the thickened cell wall. So this is a giveaway that this is Collenchyma, unevenly thickened cells, thickened at the corners predominantly. Sitting underneath that, we have cells that are roughly isodiametric in shape but are green in color. That's a giveaway. You're looking at Chlorenchyma. Below that, isodiametric cells, very large cells, but no chloroplasts. That is your parenchyma. There is more parenchyma here in the pith. Okay, and obviously all in between. That's all parenchyma. If we look now at a vascular bundle, in the middle, going back to our meristematic tissue, there is your vascular cambium, which as it differentiates, if it differentiates up, that will become phloem. As it differentiates downwards, that will become your xylem. Sitting above that, you can just see some of these cells have little dots in them. Okay, those are your sieve plates. And if you look even closer, you might be able to identify some of your companion cells, like just there is a companion cell. So that's a giveaway that you are looking there at phloem. These large circular structures, that's a dead giveaway that you are looking at xylem. The newest xylem is actually the one that is the largest. Okay, those are your metaxylem. You don't need to know meta versus protoxylem, you just need to know xylem. But I'm informing you that these big ones, these are actually your newest ones. These ones were formed when this plant was still much, much younger. Because obviously as your cambium starts to create new cells, those new cells are going to push into this space here. And so the newest xylem are these ones. Your newest phloem are equally those ones that are nearest to the cambium, because as these cells multiply, they're going to push into the phloem. And so the newest phloem will be over here, the youngest phloem. Protecting your xylem and your phloem, you can see here, these cells have evenly thickened cell walls. And so this is your Sclerenchyma. So there's a cap of Sclerenchyma which protects the plant from injury coming from outside, physical injury. So those are some of your tissues, looking at them in situ inside your sunflower stem. If we look at the same thing in a monocotyledon, just zooming in a little bit more, makes it a little bit easier possibly to see a number of vascular bundles. So again, on the outside is your epidermis with a cuticle. Underneath that is Collenchyma. How do we know? We know because the black lines seem bigger and thicker in between here, and that's because of your unevenly thickened cell walls. These large circles, dead giveaway. These are xylem vessels. Sitting above your xylem vessels are your phloem cells. Again, you can identify them. They've got dots. Looks like little kind of speckles or a badly printed picture almost. Okay. Equally, you've got lots of large cells with lots of small cells mixed in between them. That's a giveaway that you're looking at phloem. And then on the outside, always on the outside, looking towards where the stem is, you've got your Sclerenchyma cap, or a fibrous sheath, as it's sometimes called. Okay. And again, how do you identify that? Because the black lines in between are very thick, but unlike your Collenchyma, those black lines will be the same thickness throughout. So those are your different permanent tissues.